So in this video, we're going to talk a little bit about reabsorption in the kidneys, and that involves refreshing your mind on some anatomy that we've already talked about. So I am going to draw for you a representation of the glomerular capsule, or Bowman's capsule. And you guys have learned already that it's continuous with a structure that's known as the renal tubule. And the reason that this particular area is important is because it connects up with the glomerulus. So I'm drawing a really simplified representation of the blood vessels now. So here's our glomerulus, efferent arteriole, and you guys know that that becomes continuous with the peritubular capillary. So we have the peritubular capillary here in red, and over here the renal tubule. And the reason that this particular area is important is because it's where filtration takes place. So blood is moving through this blood vessel here into the glomerulus, and once that blood enters into this area, we have filtration occurring, right? And that means that the stuff that was in the blood, at least the stuff that's small enough to fit, is going to move out of the glomerulus and into that glomerular capsule, or Bowman's capsule. So I'm going to draw you a representation of some of the things that get filtered out in this area. So one of the major things that gets filtered out is water, and I'm going to use just some little blue dots to represent water that has moved out of the blood during this process of filtration and is now in the glomerular capsule. Another thing that gets filtered out, and this is actually the primary solute that's in the filtrate, are sodium ions. So I'm going to use little stars to represent sodium ions that have been pushed out of the blood and moved into this glomerular capsule, or Bowman's capsule, through the process of filtration. And then of course we've got all of the other things that are in the blood that make their way out. So this would be things like glucose and amino acids and other ions that make their way out of the blood. And I'm going to refer to this as just everything else, okay? Because we'll talk about it a little bit later. So again, things like glucose, amino acids, and other things. And so that's the process of filtration. And of course, once you have all of this stuff moved out of the blood, what happens next is it starts to move down the renal tubule. So the sodium ions, the water, everything else that was pushed out of the blood starts to move down the renal tubule. And of course, the renal tubule is adjacent to the peritubular capillary. So what I'm going to do now is I'm going to take this little area of anatomy where we have the renal tubule and we have the peritubular capillary adjacent to each other, and I'm going to draw you what these would look like much more highly magnified, so we can now start talking about this process of reabsorption. Because remember, reabsorption is what happens when stuff here in the renal tubule gets taken back up into the peritubular capillary, so we get it back into the bloodstream and we conserve it within the body. So if we go over here, okay, so again, here is that renal tubule. So that area that we were looking at previously, just a little bit more highly magnified. And right next to that, we've got the peritubular capillary, so that blood vessel. And in this process of reabsorption, what we're trying to do is take this stuff that was pushed out of the blood during filtration and again, move it back over here into the peritubular capillary. And of course, the peritubular capillary itself, okay, is made up of cells. So the wall of the peritubular capillary is cells, and I want to draw those in because they are really important to the functioning and how this reabsorption process actually occurs. So I'm going to put some nuclei in there just to make it clear that we're talking about cells that make up the wall. And I'm going to draw some cells down in here as well. Okay, they're little nuclei because they make up part of that renal tubule wall. So anything that's going to be reabsorbed is going to have to move from this area where the filtrate is, across this cell membrane, through the cell, across the other cell membrane, into this space here between the renal tubule and the peritubular capillary. And from there, it's going to have to move through the wall of the peritubular capillary across that membrane to actually get into the blood. And then of course, once it's in the blood, blood is flowing and it's going to whisk it away in this direction and move it away from the renal tubule. So I want to draw that stuff in that we said is part of the filtrate. We've got lots of sodium ions, right? That's our main solute that's in the filtrate. So I'm going to put lots and lots of those. And we have a lot of water molecules in there as well. So water that's just left the blood and it's now in the renal tubule of the kidneys. And then of course we have the everything else, right? The glucose, the amino acids. There's some of those things that are within this particular area as well. Now, one thing that you should know is that all of these cells that make up the renal tubule wall on what's known as their luminal surface, okay, which is this surface here that's adjacent to the lumen or to the opening of that renal tubule. They have sodium channels. And you guys at this point should be familiar with what sodium channels are. These are little openings through a cell membrane that's going to allow specifically sodium to cross through them. So what happens because we have these sodium channels is that sodium starts to move through them into the cell. So we get sodium moving out through those sodium channels into these cells. And then we've got another thing over here on this surface and on what's known as the basolateral surface of these cells. So on the basolateral surface, we have some sodium-potassium pumps. And you guys may remember that sodium-potassium pumps use ATP to actually take sodium and move it in one direction, take potassium and move it in the other direction. So as soon as we get a sodium ion in here, what's going to happen is that sodium-potassium pump is going to take it and it's going to push it into this space in between the renal tubule and the peritubular capillary. So sodium continues diffusing through these sodium channels and we end up with a high concentration of sodium in this particular area. And you guys know anytime you have a high concentration, it's going to try to reach equilibrium. And so what happens then are these sodium ions move from this area where they're really highly concentrated in this space and they move into the peritubular capillary. The peritubular capillary whisks them away in the blood, and that's how we're going to reabsorb the sodium ions. One of the things that's really cool about this is most of this reabsorption is happening passively, and it's happening just due to concentration gradients. So initially, basically all the sodium gets filtered out of the blood, and it's sitting over here in the filtrate. On this side of the surface, we have those sodium channels, and sodium channels don't require ATP. There's no energy needed for them to work. Sodium just moves from that area of higher concentration to the area of lower concentration inside of the cell through that process of diffusion. And then here's where we've got some active transport happening, right? We've got those sodium-potassium pumps, and they do use ATP in order to work. So as soon as we get a sodium ion over here, that pump grabs it and moves it into this area. That prevents us from ever coming to equilibrium between here and here. And because we don't come to equilibrium, sodium can continue to move across that membrane until we have absorbed basically every single sodium ion. And then every single sodium ion that gets diffuses across moves into this space. We get a high concentration here. That's going to drive it just passively through passive transport, no ATP needed, into the peritubular capillaries. And we don't come to equilibrium. We maintain a concentration gradient on these two sides of the membrane because remember, as soon as sodium's here, it's whisked away. And that keeps this area at a very, very low sodium concentration. So we're getting reabsorption of sodium, and it's happening really without the use of energy, without the use of ATP being expended by the kidneys. But here's where it gets even cooler, okay? So we have all this sodium being reabsorbed, and I want you to think about if all this sodium is being reabsorbed, how that's gonna change what the filtrate looks like as it continues to move down the renal tubule. So after a lot of our sodium is absorbed, okay, we're still gonna have this everything else, and we're gonna have some water molecules, a lot of water molecules because we haven't reabsorbed any of those yet. So I'm gonna start drawing those in. So here's all our water molecules. We'll still have some sodium here, right? But most of our sodium is already making its way out of the renal tubule. So we end up with a situation where we've got a high concentration of sodium here, a very low concentration of sodium here. Sodium can't move backwards to reestablish equilibrium because sodium-potassium pumps don't allow that to happen. They're not channels. They pump sodium in a specific direction and they're not capable of moving it back in the other direction. So we've got a concentration gradient. The sodium can't move back, and you guys know that whenever you have a concentration gradient and the solute can't move, that what happens is water starts to move. So we get this concentration gradient, and then water starts to follow the sodium. And we actually have spaces that water can move through, and we get water moving inside of these cells. It builds up here. We've got sodium on the other side of that cell, and that sodium is going to pull the water. Then we get water building up in here that moves into the space. So now we have basically our water following sodium. It's moving passively. We're not having to expend ATP to pump it. It's just following that sodium to try and maintain a concentration gradient. So now I want you to think about this again, okay? We've reabsorbed the sodium. We're reabsorbing the water. How's that going to change what the filtrate looks like? Well, now we're going to get a situation like this, okay, where we've still got that everything else, the glucose, the amino acids, etc. We might have a sodium ion or two, okay, a little bit of water, but most of our water, right, is making its way out. And so we have a very high concentration of water here, a very low concentration of water here. So once again, we've got a concentration gradient. Now what's going to happen is this everything else is going to start to move to try and reestablish equilibrium. So it's just going to follow the water, trying to maintain an equilibrium. And so now we're getting our glucose, our amino acids, those things being reabsorbed as well without the use of ATP. We didn't have to pump them because we pumped sodium just right here, mostly got it to move across through diffusion. That set up a concentration gradient with water, which caused water to move across through osmosis. Diffusion of water is osmosis, right? And then that set up a concentration gradient with water, and it pulled those everything else, right? All those other things, it pulled those across as well through a passive transport process. So absolutely amazing engineering that's going on in the kidneys. My sister's a chemical engineer. I explained this process to her one time. She was blown away because it's cool. We're moving all of these things and we're doing it without the use of energy for the most part. The only place we're using energy is right here with those sodium potassium pumps. And so that makes this a very, very efficient, very, very effective process that's happening within the body. And that, in a nutshell, is reabsorption and how sodium drives the reabsorption of most of the things that are in the filtrate.